Friends, hello, welcome to my channel once again. Now this is a tutorial video for hormone action. A very short video in fact. Now here we see a picture. Actually this is a action of any proteinaceous or amine hormone. Let this be the hormone. The one that I am painting over here, let this be the hormone. This is the receptor of the hormone present on the cell membrane. Now here, with the help of ATP, the enzyme over here is adenylate cyclase or adenyl cyclase. It is converted into CAMP, means cyclic adenosine monophosphate. From there it reaches the nucleus. Within the nucleus there is the DNA, and this DNA is activated with the help of CMP and it releases the receptors. The receptor means the hormonal actions, whatever it is. Let it be insulin over here. So as far as the knowledge goes, the action of the insulin starts over here. This kind of hormone action is seen in protein or polypeptide hormones like insulin, ADH and TSH. Now they are water soluble and unable to pass through the lipid bilayer of the cell membrane. The steps over here are, the first step is hormone binds to extracellular receptors, as you see over here. This is the extracellular receptor. Example is given here. After that, the production of adenylate cyclase occurs, then production of cyclic AMP or calcium ions occur. As a result, cyclic AMP enters the cell as a second messenger, or calcium may be the second messenger, and after that cascade of reactions take place, forming a physiological response, means ovarian growth. If it is FSH, then the resultant is ovarian growth. We come to another thing, means what are second messengers? Second messengers are, one is IP3, another is DAG or DG. We will study over here. Now let this be insulin. This is also the extracellular receptors. It contains 4 subunits, 2 alpha and 2 beta. The alpha subunits are insulin binding subunits, which are at the insulin binding site of the membrane. Now it is combined with the beta subunit, which is a tyrosine kinase domain, means beta is the tyrosine kinase enzyme, spanning the entire membrane. Now this is the inside of the cell. The inside of the cell releases phosphates. These phosphates, they in turn, they attract the insulin receptor substrate. Now here we come to the steps. Here beta subunits add phosphate groups of specific tyrosine residues, due to which the transducer G protein activates phosphodiesterase enzyme that makes the enzyme phosphatidyl inositol 4,5 biphosphate or PIP2. It is divided into two, means into a pair of mediators. One is inositol triphosphate, as I told you, IP3. Another is DAG or DG, which is diacyl glycerol. IP3 triggers the release of calcium ions from intracellular endoplasmic reticulum. And on the other hand, DG activates the protein kinase C enzyme, which further activates the pyruvate dehydrogenase, and therefore the action begins. Means insulin has its action in the nucleus of the cell and then comes to the nucleus, out of the nucleus. So this is the video. I think you will get enough knowledge from this. So best of luck to all the examiners, all the examinees in fact. Thank you.